All right, so in the last section, part 2, we were talking about Mendel and how he figured out how pea plants pass on their genes and Punnett squares, and figured out that organisms have 2 alleles for every gene, and it's how those alleles interact that can predict what they're going to look like, or what, if they're lactose intolerant or what have you. The examples we were showing you guys were an example of a dominant recessive inheritance pattern, and I want to explain that in a little bit more detail right now. There's a whole bunch of different inheritance patterns we're going to look at. The first one, the one that Mendel figured out first, was this dominant recessive one. So let's take a look. Okay. If a genotype has a dominant allele, it will always be expressed. Now, we did this one already in the vocabulary section. So dominant, we portray with a capital E. In this, if we're doing ears, right? We'll do capital E. Okay. For ears. And we'll do, so dominant, be the big letter, right? Bigger folks tend to be more dominant. If a genotype has a recessive allele, it will only be expressed if it's homozygous recessive. Now, homo means same, right? We've seen that before. Homo means same, hetero means different. So the recessive allele will label with a little e, because it's a smaller one. And the only time you see that is when there's 2 little e's. I like to think of this like playground politics, right? You guys were all in elementary school at some point, little kids on a playground somewhere. If you were, let's say, 5th graders and kindergartners, happen to be on the playground at the same time, unsupervised. If it's all 5th graders, obviously the 5th graders get to choose what they do. They all get to play Foursquare, right? No big deal. If the kindergartners are out on the playground all by themselves, they get to play Foursquare if that's what they want to do, or go swinging or whatever. But if you have 5th graders and kindergartners on the playground at the same time, who gets their way? Who gets to choose what's happening? It's the 5th graders. The 5th graders are like the dominant big E alleles in this case, and the kindergartners are like the recessive little E alleles. Okay, dominant recessive, you can think of like playground politics in that way. So that's what we saw happening. The question is, why does that work? It's one thing to know that it happens. It's one thing to know that if you drop something, it falls. It's another thing to know why it does that. So let's take a look at that flower gene color, gene for flower color from above is a good example of this. All right, so we got that purple flower. Okay, one allele codes for making a protein that produces a purple pigment. So as we know before, DNA is simply a spelling book for putting amino acids together in the right order to make a protein. That protein then carries out a particular function. Could cure action in your blood, it could fight COVID for you, or it could just be a pigment color or make a pigment color, right? Well, in this case, for the purple flower, that allele, that gene for flower color, actually makes a purple pigment. Okay? Now, on the other hand, the other allele in this case does not make a functional protein. It makes no color. So something happened in the past of the ancestors of these plants where something happened to that color gene that made it inoperable, right? It wasn't able to make the full pigment protein. So that plant, or that piece of DNA in that plant, is not able to make a pigment. And in the lack of pigment, in this case, the underlying color shows through, which is white. Okay, so it's not that it's making a white pigment, it's that it's not making a pigment. So let's take this a little further. So if you get 2 alleles that both make purple pigment proteins, okay, 2 alleles both make purple pigment proteins, which we will designate with big P, or we can use our 2 purple allele markers like we did before. What are you going to get? Then you have a purple flower. Also, if you get 2 alleles that both cannot make a functional pigment, little p, which we'll designate with the white ones, right? Well, we get a white flower because there's no pigments being produced, so the white shows through. No big deal. That's pretty obvious. If you got all proteins making pigments, you're going to have the color. If you got all proteins that can't make the pigment, no color. So what happens when you have one of each? Now, if you have two alleles, but they are different, or what we called before heterozygous, hetero for different, one that makes a purple pigment, big P, and one that makes no pigment, little p, your genotype now is big P, little p, or a purple and a white. That is your gene type, right? Your genotype. So how does that affect your phenotype, what you physically look like? So my question to you is, what pigment will this flower produce? Well, the DNA strand that's got the allele for color that makes purple pigment will still make purple pigment. And the chromosome that's got the gene that can't make a pigment because something's messed up there isn't going to make a pigment. So in other words, you're still making a purple pigment. If you're still making a purple pigment, your phenotype is purple. So the way dominant and recessive works is your dominant allele is the one that makes the protein. So it still does what it's supposed to do. And the recessive allele, the recessive version of that gene, is not producing a functional protein at least. It might be producing some protein, but it's not functioning in a way that affects what's going on, in this case, color, or in the case of hair loss or whatever it might be. I'm not even sure if that's a dominant recessive trait. So that is dominant recessive. The dominant makes the protein. The recessive doesn't make a functional version of the protein, which is why the only time you see the lack of the protein, the recessive trait, is when if you have two alleles that both don't make the protein, two recessive, little p in this case. All right, so now let's do this with Punnett squares and just illustrate this a little bit. So first step in a Punnett square is to come up with a parent cross. So right here, I'm just going to turn this a little bit so you can see me. So we got the parent cross, one parent here, that'd be a purple flower, that'd be another purple flower parent, both homozygous dominant. Homo for same, dominant for having the big P, the dominant trait. The next thing that happens with that flower in this case, right, is you go through meiosis. You make the gametes, you make the sperm and the egg. So let's do that. So this one, how many alleles can be in a sperm cell for a particular gene? Just one. So half of them are going to go in one sperm cell, and the other half are going to go in the other sperm cell. And granted, these plants are not making sperm cells with tails. I'm drawing this because it's evocative of a gamete, a sex cell, that everybody understands and recognizes. So this visual should hopefully help you with the understanding, remembering it. Now, here, the female plant is going to make eggs, and they're little peas. I'll put a little line over it because the big pea and a little pea look a lot alike, especially the way with my handwriting. So you put that little line over it, it helps you to know that it's a lowercase. All right, so we've got the parent cross here. We've separated what we call separating the gametes, or the egg and sperm next. That's the second step. And the last step is to put those in the Punnett square. Now, key factor here is you got to put the gametes from the same parent on the same side of the Punnett square. So I'm going to put the eggs on top, one egg on top, I got to put the other egg on top. Okay? If I put one sperm on the side, I got to put the other sperm on the side. I can't go mixing that up. All right, they've got to be the same. These are not little peas. What am I doing? Those are supposed to be big peas. I'm talking, but not paying attention. So we got big P across a big P, big P. So if that big P egg is fertilized by this big P sperm, we just bring them over. A big P from the sperm and a big P from the egg. What color is that going to be? It's making purple pigment, right? So it's going to be purple. If this sperm, bring it over to this square, fertilizes this egg, if that's the egg that's ovulated, we're going to get a big P and a big P. The genotype is big P, big P. That means it's making two dominant, so it's making the protein. It's going to be purple. Same thing here. This egg with this sperm, that's two big P's again, purple. And this one over here, we just bring it down, and we got purple. So if we mate those two plants, two homozygous dominant purples, remember homozygous, same, dominant because they're capitals, making the pigment. we are going to get all the kids are going to be the same. They're all going to be big P, big P. All right. Let's take a look at the next one. Let's take a big P crossed with a little p. So a homozygous dominant with a homozygous recessive. So if we separate the gametes again, we're going to get a big P in that sperm, a big P in this sperm. And these eggs are going to be what I drew last time, little p's. So that's the parent cross. Separate the gametes. Put the same parent on the same side. I'm not going to draw the egg and the sperm on this one. And maybe I should. I'll just draw them. And then we got the big P sperm over here. And if we go ahead, if that sperm happens to fertilize this egg, if that's the chance of occurrence, we're going to get a big P, little p. So this is heterozygous, right? Different. And in this case, it's we got a 5th grader and a kindergartner. Who wins? The 5th grader. This is purple. We're making the purple pigment, so we're purple. And if you take a look, all the other ones are going to be the same. This is a big P over here, and this little p down here, purple. Little p from the mother, big P from the father, purple. And the last one is the same combination. Pretty straightforward. As well, we wound up with all heterozygous kids. This is what was happening in that example I showed you from Mendel, where he took the purple plant, what we call a true breeding purple plant, a purple plant that came from generation after generation after generation of plants that just had purple plants, and he crossed it with a little p, or what he would have called a true breeding white, came after generation of white, so it was likely little p's. When he did that, he saw all the kids were purple. We are going to do a big P, little p, cross with a little p, little p. So 2 eggs and 2 sperm. We separate the gametes. There's the little p's from the mom, a big P from the dad, and the little p sperm from the dad. If we do that, big P from the dad, little p from the mom, purple. Big P, purple. Little p, right? Little p. That is white. 2 recessives. Same thing here, white. So if you did this mating, you would expect to have half the kids purple, half the kids white flowers. And lastly, let's take a look if we just did all recessives. I imagine you can figure this one out. These are little p sperms and little p eggs. And eventually, you don't have to draw eggs and sperm, right? You can just do, we have little p sperm, little p eggs, and then these are all the same. So we know it's pretty easy when they're all the same and they're homozygous like that. They're all white kids. So if you do 2 recessive plants mated, all the kids are going to be recessive. So it's kind of like the thing if you mate, if 2 light-eyed people, blue, green-eyed people mate, light eyes is recessive. All of their kids are going to be light-eyed. Okay, and we'll see that in an example later on. And that'll do it for looking at basically how to do a Punnett square, how Mendel figured this stuff out, and how the dominant and recessive inheritance pattern works. And we'll see you later.